For decades, serial killers haunted the shadows, leaving a trail of fear and unanswered questions. Police struggled to track these elusive predators, hampered by limited forensic tools. But the landscape of crime-solving changed dramatically with the advent of DNA profiling. This groundbreaking technology, like a beacon of hope, offered a powerful new weapon in the fight against violent crime. It provided investigators with an unprecedented ability to identify criminals through their unique genetic fingerprint. DNA profiling, often compared to a molecular fingerprint, quickly became an indispensable tool for law enforcement agencies worldwide. It provided irrefutable evidence, linking perpetrators to crime scenes and victims with astonishing accuracy. The ability to analyze trace amounts of biological material such as blood, saliva or hair revolutionized investigations, bringing long-sought justice to countless victims and their families. The impact of DNA profiling extended far beyond individual cases. It fundamentally reshaped the criminal justice system, ushering in an era of forensic science never seen before. The ability to revisit cold cases, exonerate the wrongly convicted, and identify perpetrators years, even decades, after their crimes had a profound impact on society's perception of justice and accountability. At the heart of this revolutionary technology lies DNA, the very essence of life. This intricate molecule, residing within the nucleus of every cell, holds the genetic blueprint that makes each individual unique. Like a coded language, DNA carries the instructions for our physical traits from eye color to height, and this uniqueness is the key to DNA profiling. Within the vast expanse of the human genome, scientists discovered specific regions called short tandem repeats, or STRs. These STRs are short sequences of DNA that repeat a variable number of times in different individuals. It is the variations in these repeats that make each person's DNA profile unique, distinguishing them from every other individual on Earth. DNA profiling capitalizes on this variation. By analyzing a specific set of STR markers, scientists can create a DNA profile, a unique genetic fingerprint that is highly unlikely to be shared by anyone else, except for identical twins. This profile becomes a powerful tool in criminal investigations, providing compelling evidence to link suspects to crime scenes or exonerate the innocent. The journey of a DNA sample, from a crime scene to a courtroom, is a meticulous process involving forensic scientists and law enforcement. It begins at the crime scene, where professionals collect DNA evidence. Samples are packaged and transported under strict protocols. At the lab, DNA is extracted and analyzed. The DNA is isolated and amplified using PCR. Electrophoresis separates DNA fragments, creating unique patterns. These patterns are compared to known profiles to identify suspects. Section 4 DNA on trial cases cracked by genetic clues. DNA profiling has been crucial in solving complex crimes, especially those involving serial killers. Ted Bundy, the infamous serial killer, was convicted with the help of DNA evidence. DNA evidence secured his conviction for the murder of Lisa Levy in Florida. Colin Pitchfork was the first person convicted of murder based on DNA evidence. His DNA matched samples from 2 crime scenes in Leicestershire, England. These cases highlight the power of DNA profiling in criminal investigations. Section 5. The double-edged sword, limitations and ethical concerns of DNA profiling. DNA profiling is powerful but has limitations and ethical concerns. Errors in testing, especially with degraded samples, can lead to wrongful convictions. Privacy and genetic discrimination are major concerns with DNA databases. Balancing benefits and individual rights is a challenge for policymakers. Despite concerns, DNA profiling revolutionizes forensic science and justice.